At Exploratorium, exhibit ideas are inspired by observation and curiosity. By partnering with artists and other thinkers who look at the world in a unique way, and also by staying current with innovations in science. This is what happened when stem cell researchers from the Gladstone Institutes and UCSF collaborated with Exploratorium.
We previously had been working with mouse cardiac myocytes, and we were really interested in the human cells, and we knew that human iPS cells was something happening, was very active in research labs.
Stem cells are cells that can turn into any other cell in the body. An embryonic stem cell, like a fertilized egg, can become absolutely any other kind of cell in the final organism. Scientists have learned to make human stem cells, called iPS cells, from adult cells, getting around the ethical dilemma of using embryonic cells. Stem cell researchers are studying several areas that hold great promise for advancing human health and longevity. Bruce Conklin's lab at Gladstone is growing tissue to study diseases and targeted medicine. The field is on the cutting edge, and the Gladstone Institutes are some of the most advanced. The idea for the responsive heart cells exhibit, which is part of a thematic set of new exhibits called Cells to Self, was born from an ongoing cross-pollination between Exploratorium biologists and scientists from the Gladstone Institutes. Alexandre Rubiero was a scientist at Gladstone when he attended a retreat at Exploratorium, where he heard Exploratorium staff scientist Christina Yu talk about the advanced cell culture capabilities of her staff and biolab. It gave him the idea for an exhibit.
At the time I was working with those types of cells, and I was doing basically what the exhibit does. I was pacing cells at specific frequencies. What if, I could pace them at my heartbeat, at my heart rate? It was always something that I always thought.
The cardiomyocytes in the exhibit are beating human heart cells, transformed from adult human skin cells at Gladstone.
I'm also a huge fan of the Exploratorium. My wife loves the Exploratorium, so we would go there quite often. And it's very interactive. So a lot of the exhibits in the Exploratorium, you do it yourself. It's by putting yourself into the piece, you'll learn.
Letting visitors control the beating of human heart cells with signals from their own hearts resonated with everyone who heard it. But something like this had never been tried in a museum before. Could these cells be brought to the floor and survive Exploratorium's eager visitors? The scientists didn't know either.
The researchers are growing iPS cells and making new types of cells and keeping them alive for usually the length of their experiments. Some experiments are days long, sometimes they're weeks long. Occasionally they're a little bit longer, but most of the research is days to weeks. We have a limited access to the cells. We have a limited supply of cells, so we're trying to make them last for 3 to 4 to 5 months in culture.
While the Exploratorium is expert at building exhibits, evaluating, and iterating, the required biological expertise was pushing the boundaries of exhibit development into new territory. Director of Living Systems Christina Yu began to assemble a small but experienced team around Angela and her biological expertise for this groundbreaking exhibit. New media developer Chris Cerrito was chosen to work on the project for his combined hardware and programming expertise.
And I honestly, when it was first talked about, had no idea what anybody was speaking about because I didn't have a clue what a cardiac myocyte was. I'm not a biology person. So I kind of remember sitting there in a daze for a while until somebody explained to me, like, oh, we're going to make people interact with human heart cells. And that got me excited. And Angela had the task of keeping these cells alive, which is crazy. And honestly, I'm really glad that wasn't my part. She's amazing. What we started with was figuring out what's the best and easiest way to accurately get a visitor's heart rate. So at the very beginning of this project, we did a lot of rapid prototyping with different kinds of sensors, some that were finger clips, some that you just put your hands onto. We ended up going with this sensor right here, which involves 2 electrodes, which the visitor grabs.
The resulting electrical pulse is immediately sent to the cardiomyocytes under the microscope. The cells receive the electrical signal and simultaneously start beating at the visitor's rhythm. Text and graphics are tested and modified along with each iteration of the exhibit. The team tests new things out on each other and with other Exploratorium staff. Special groups are often arranged.
Bruce Conklin's lab came here on a retreat, and we set up the prototype of the exhibit for them to use, and they were really excited about it. Some of the people who thought it wouldn't work were especially surprised because it did work and it responded to them.
Figuring out what works with regular visitors is a process Exploratorium staff have been doing for almost 50 years.
That's nice.
But the central phenomenon of this exhibit, controlling a dish of human heart cells with your own heart, is new territory, requiring a level of expertise not often found at many museums. The results may have far-ranging effects.
Basically, what's the role of beating on the physiology and the health of the heart's function? And probably a system like the one developed at the Exploratorium could help scientists understand that.
When we got the cells, we weren't sure how they were going to behave. Like, no one had used these cells for those purposes before, so we were like, "Wow, this is crazy." But you know, we just decided, let's just give it a go. Let's give it a shot. Let's do it, and it's working.